In this lecture, we're going to discuss the fascinating topic of animal reproduction and development. Remember that reproduction is very important for natural selection. It's the way that individuals pass on their favorable genotype to the next generation. Therefore, traits that are directly involved in reproduction are under strong natural selection. Such strong selection can drive a diversity of reproductive anatomy, physiology, and behavior. The particular way in which animals reproduce varies among species, from the familiar to what might strike us as truly bizarre. To take just one of many examples, soon after it is born, a male anglerfish finds a female anglerfish, bites her skin, and fuses with her. His body atrophies, leaving the male reproductive organs physically attached to the female and allowing her eggs to be fertilized when she is ready. So let's get started exploring this topic in more detail. Organisms have two potential ways to reproduce. One is the production of genetically identical cells or individuals called clones. This form of reproduction is called asexual reproduction, and it occurs in a wide variety of organisms. Bacteria and archaeans reproduce by binary fission, a form of cell division in which the genome replicates and then the cell divides in two. The result is two cells that are either identical to the parent cell or nearly so, differing only because of chance mutations. Saccharomyces cerevisiae, shown on the upper right, which is the yeast used to make bread and beer, is so named because of its ability to produce new individuals by budding. Budding is forming a bud or protrusion on an organism that eventually breaks off to form a new organism that is smaller than its parent. Certain molds, algae, worms, sea stars, and corals reproduce by fragmentation. Fragmentation is a process in which new individuals arise asexually by the splitting of one organism into pieces, each of which develops into a new individual. Parthenogenesis occurs in invertebrates such as bees, other insects, and crustaceans. It can also occur in vertebrates, including some fishes and reptiles, like the Komodo dragon seen to the lower right. Parthenogenesis is a form of asexual reproduction where females produce eggs that are not fertilized by males, but divide by mitosis and develop into new individuals. The second way that organisms reproduce is by combining a complete set of genetic information from two individuals to make a genetically unique individual. This form of reproduction is called sexual reproduction and occurs only in eukaryotes. At its core are two basic biological processes, meiosis and fertilization. Meiosis is a form of cell division that halves the number of chromosomes. Fertilization involves fusion of specialized cells to restore the original chromosomal content. Meiosis consists of one round of chromosome replication, followed by 2 rounds of cell division, resulting in 4 cells that each have half the number of chromosomes as the parent cell. The resulting cells are haploid and are called gametes or spores. The products of meiotic cell division, the 4 haploid gametes, are each genetically unique and represent a mixture of the parental genetic makeup. This process is covered in more detail in the Bi 204 course of this introductory biology series. Fertilization involves the fusion of 2 gametes, one from each parent. The result is a zygote. The zygote divides by mitosis and develops into an embryo, an early stage of multicellular development. Many species produce 2 types of gamete that differ in shape and size. The smaller male gametes are called spermatozoa or sperm. The larger female gametes are called ova or eggs. Most organisms that reproduce asexually are also capable of reproducing sexually. As one example, the freshwater crustacean Daphnia reproduces asexually by parthenogenesis in the spring, as shown in the lower left. This is a time when food is plentiful, so perhaps they engage in asexual reproduction because it is a rapid form of reproduction that allows them to take advantage of the abundant resources. Later in the season, when conditions are less favorable because of increased crowding and decreased food and temperature, Daphnia reproduce sexually, mating to produce a zygote that starts to form an embryo. At an early stage, development is arrested and metabolism slows dramatically. The embryo forms a thick-walled structure called a cyst, which enables it to resist freezing and drying and therefore last through the winter. Asexual reproduction has some advantages compared to sexual reproduction. For one, asexual reproduction does not involve finding and attracting a mate, which takes considerable time and energy. Second, asexual reproduction is rapid, which allows organisms to increase their numbers very quickly. Let's consider rates of population growth for organisms that reproduce asexually and those that reproduce sexually. In asexual organisms, all offspring can produce more offspring, allowing for exponential population growth if conditions are favorable. In sexually reproducing organisms, only females produce new offspring. The consequence of this difference is that an asexual group can have a much greater rate of population growth compared to that of a sexually reproducing one. The British evolutionary biologist John Maynard Smith called this effect the twofold cost of sex, and it is one of the key disadvantages of sexual reproduction. Despite these costs, sexual reproduction must confer some adaptive advantages. Hypotheses for these advantages include that 1. It allows organisms to adapt faster than asexual ones because rare beneficial mutations that arise in different organisms can be brought together, increasing the overall fitness of the population. 2. Sexual reproduction allows a population to purge itself of harmful mutations more quickly than could a population of asexual individuals. And 3. Sexual reproduction may be a mechanism of parasite defense. These different explanations for the prevalence of sexual reproduction are not mutually exclusive. It may be that several mechanisms work together, or that different ones are important for different organisms. Egg and sperm require a wet environment to survive. In aquatic organisms like fish and amphibians, eggs and sperm can be released directly into the water in external fertilization. External fertilization is a chance affair. Somehow, sperm must meet egg in a large environment. Animals that release gametes externally, therefore, have developed strategies to increase the probability of fertilization. One strategy involves releasing large numbers of gametes. Externally fertilizing animals may also come close together physically to improve chances that the sperm will fertilize the eggs. Internal fertilization, in which fertilization takes place inside the body of the female, is an adaptation for living on land. Reptiles, birds, and mammals all use internal fertilization. However, this mode of fertilization is not exclusive to land dwellers. Marine mammals, such as whales and dolphins, use internal fertilization, as do the land-dwelling ancestors from which they evolved. Even some fish, such as guppies, reproduce by internal fertilization. The two modes of reproduction, external and internal fertilization, are associated with a host of other behavioral and reproductive differences. One key difference is the number of offspring produced. External fertilization is generally associated with the release of large numbers of gametes and the production of large numbers of offspring, each of which has a low probability of survival. Animals that use internal fertilization typically produce far fewer offspring and invest considerably more time and energy into raising those offspring. As shown here, these two strategies represent two ends of a continuum of reproductive strategies that was first described by the American ecologists Robert H. MacArthur and E. O. Wilson in 1967. Organisms that produce large numbers of offspring without a lot of parental investment are called R strategists, and those that produce few offspring but put in a lot of parental investment are K strategists. According to this model, the environment places selective pressures on organisms to drive them in one or the other direction. R strategists evolve in unstable, changing, and unpredictable environments where there is an advantage to reproducing quickly and producing many offspring when conditions are favorable. K strategists often evolve in stable, unchanging, and predictable environments where there tends to be more crowding, larger populations, and intense competition for limited resources. So traits such as increased parental care and few offspring are favored. Parental care begins early in development in the provisioning of nutrients for the developing embryo. The eggs of both terrestrial and aquatic animals that are externally fertilized have a yolk that provides all the nutrients that the developing embryo needs until it hatches. The movement onto land was associated with important changes in the anatomy of the egg. No longer could the developing embryo depend simply on diffusion of key substances into and out of a watery environment. A key innovation for terrestrial life is the amnion, a membrane surrounding a fluid-filled cavity that allows the embryo to develop in a watery environment. Reptiles, birds, and mammals are known as amniotes to reflect the presence of this key membrane. Let's take a more detailed look at the amniotic egg. First, we have the amnion, a membrane surrounding a fluid-filled cavity that allows even a terrestrial animal's embryo to develop in a watery environment. A second membrane, called the allantois, encloses a space where metabolic waste collects. A third membrane, called the chorion, surrounds the entire embryo along with its yolk and allantoic sac. The yolk sac, amnion, allantois, and chorion are extra embryonic membranes, sheets of cells that extend out from the developing embryo. The embryo is further protected in many species by a hard shell that prevents drying out and facilitates gas exchange with the surrounding environment. Animals that are internally fertilized can either lay eggs, give birth to live young, or combine both strategies. The strategy of laying eggs is called oviparity, directly translated as egg birth. Oviparity is used by fish and amphibians, as well as reptiles, birds, and most insects. There is very little, if any, embryonic development inside the mother, so all the nutrients for the developing embryo come from the yolk. Animals that give birth to live young are viviparous. The embryo develops inside the mother, and nutrients are obtained directly from the mother. A more rare type of birth is ovoviviparity, in which embryos develop inside eggs that are retained within the mother's body until they are ready to hatch. The mother gives birth to live young that have hatched from the internal eggs. Some shark species adhere to this reproductive strategy along with other animals. Humans, like all mammals, are amniotes. They reproduce sexually using internal fertilization and are viviparous. In the human male reproductive system, gametes called spermatozoa, or sperm, are produced in the two male gonads, or testes. Sperm are haploid cells with a unique shape and properties adapted for their function of delivering genetic material to the egg. They have a small head containing minimal cytoplasm and a very densely packed nucleus. The head is surrounded by a specialized organelle, the acrosome, that contains enzymes that are used by sperm to transverse the outer coating of the egg. Sperm have a long tail, or flagellum, which moves the cell by a whipping motion powered by the sperm's mitochondria. The male testes are located outside the abdominal cavity in the scrotum. This location is critical for the production of sperm, which requires cooler temperatures than are found in the abdominal cavity. Sperm are produced within the testes in a series of tubes called the seminiferous tubules. This is where diploid cells undergo meiosis to produce haploid sperm. When sperm are first produced, they are not fully mature. They are unable to move on their own and incapable of fertilizing an egg. They mature as they move through the male reproductive system. From the seminiferous tubules, sperm travels to the epididymis, where they become motile. This is where sperm are stored prior to ejaculation. From there, sperm enters the vas deferens, a long muscular tube that follows a circuitous path, starting off in the scrotum, passing into the abdominal cavity, running along the bladder, and then connecting with the ejaculatory duct. The two ejaculatory ducts merge at the urethra, just below the bladder. The urethra is used by both the urinary and reproductive system in males. The male reproductive system is well adapted for the production, storage, and delivery of sperm. There are several exocrine glands that produce components of semen, the fluid that nourishes and sustains sperm as they travel in the male and then female reproductive tracts. The prostate gland is located just beneath the bladder. The urethra and ejaculatory ducts pass directly through this gland. The prostate gland produces a thin, slightly alkaline fluid that helps maintain sperm motility and counteracts the acidity of the female reproductive tract. Two seminal vesicles located at the junction of the vas deferens and the prostate gland secrete a protein and sugar-rich fluid that makes up most of the semen and provides energy for sperm motility. The bulbourethral glands below the prostate gland produce a clear fluid that lubricates the urethra for passage of sperm. The urethra is enclosed in the penis, the male copulatory organ. When a male is sexually aroused, the penis becomes erect as a result of changes in blood flow. The arteries become dilated and the veins compressed. The female reproductive system produces gametes, but it is also specialized to support the developing embryo. Developing female gametes are called oocytes. The oocyte is a remarkable and unique cell. The oocyte is the largest cell by volume in the human body and is visible to the naked eye. It has large amounts of cytoplasm containing molecules that direct the early cell divisions following fertilization and food supply in the form of yolk. While a male produces millions of sperm, a female usually produces just one oocyte each month, as she will nourish and support the developing embryo if the oocyte is fertilized. Oocytes are produced in the two female gonads called ovaries. Mature oocytes are called ova and are released monthly in response to hormones. Following release from the ovary, the oocyte travels through the fallopian tube or oviduct. There are two fallopian tubes, one on each side. The fallopian tubes have feather-like projections on their ends that help channel the egg into the tube and not into the abdominal cavity. The oocyte travels from the fallopian tube to the uterus. The uterus is a hollow organ with thick muscular walls that is adapted to support the developing embryo if fertilization occurs and to deliver the baby during birth. The end of the uterus is called the cervix or neck of the uterus. The uterus is continuous with the vagina or birth canal. The vagina is a tubular channel connecting the uterus to the exterior of the body. It is highly elastic, allowing it to stretch during sexual intercourse and birth. The male testes and female ovaries not only are the site of gamete production, but are also part of the endocrine system. The master regulator of the endocrine system is the hypothalamus, which is located in the brain. The hypothalamus secretes hormones that act on the pituitary gland, which in turn releases hormones that act on target organs. In the case of the reproductive system, the hypothalamus releases gonadotropin-releasing hormone, GnRH, which stimulates the anterior pituitary gland to secrete luteinizing hormone, or LH, and follicle-stimulating hormone, FSH. These hormones then act on the male and female gonads. In males, these hormones are secreted almost continually, whereas in females, they are secreted cyclically. In response to hormones released by the hypothalamus, the testes secrete the hormone testosterone, and the ovaries secrete the hormones estrogen and progesterone. Testosterone is important during embryogenesis to direct development of the male reproductive organs. At puberty, levels of testosterone increase, leading to the development of male secondary sexual characteristics. These traits characterize and differentiate the two sexes, but do not relate directly to reproduction. Deep voice, facial, body, and pubic hair, and increased muscle mass. In females, an increase in the level of estrogen at puberty leads to the development of female secondary sexual characteristics, such as enlargement of the breasts, growth of pubic and body hair, and changes in the distribution of muscle and fat. Sexually mature females undergo a monthly cycle called the menstruation cycle, in which an oocyte matures and is released from the ovary under the influence of hormones. The menstrual cycle consists of cyclical and coordinated changes in the ovaries and uterus, timing the release of an oocyte from the ovary with the growth of the uterine lining, so it can support the developing embryo if fertilization occurs. There is a lot going on in this slide, so you may want to watch this portion a few times. The menstrual cycle has two phases. the follicular phase, and the luteal phase. In the follicular phase, an oocyte, located in a supporting shell of cells called a follicule, develops and is eventually released from the ovary. At the start of each menstrual cycle, FSH acts on a subset of follicle cells called granulosa cells. As a result, several oocytes begin to mature, but usually only one of these becomes completely mature and the others die off. The granulosa cells secrete a form of estrogen known as estradiol. Estradiol, in turn, acts on the lining of the uterus, causing it to thicken. As a result in part of rising estradiol levels, there is a rapid increase followed by a sharp decrease in the level of LH produced by the anterior pituitary gland. This surge causes ovulation and marks the beginning of the luteal phase. The follicle cells, now devoid of the oocyte, are converted to a structure known as the corpus luteum, which literally means yellow body, so called because of its appearance. The corpus luteum is a temporary endocrine structure that secretes the hormone progesterone, which maintains the thickened and vascularized uterine lining. Meanwhile, the oocyte is swept into the fallopian tube and travels to the uterus. If it is fertilized, the developing embryo implants into the uterine lining. The corpus luteum continues to secrete progesterone and is first maintained by LH and then by the hormone human chorionic gonadotropin, or HCG, which is released by the developing embryo. Eventually, the placenta takes over estrogen and progesterone production to maintain the uterine lining and stimulate growth of the uterus. If the oocyte is not fertilized, the corpus luteum degenerates, estrogen and progesterone levels drop, and the uterine lining is shed. The monthly shedding of the uterine lining is known as menstruation. The cessation of menstrual cycles, called menopause, results from decreasing production of estradiol and progesterone by the ovaries. The formation of gametes is called gametogenesis. Gametogenesis follows the same step in males and females, but there are striking differences in timing. Oogenesis is the formation of ova or eggs. The diploid precursor cells are called oogonia. These cells form during embryonic development and divide by mitosis. Some oogonia differentiate into diploid primary oocytes during fetal development. These enter the first meiotic division but arrest immediately in prophase 1, the first stage of meiosis. All these steps take place before the female is even born. At the time of birth, a female has about 1 to 2 million primary oocytes, all arrested in prophase 1 of meiosis. They do not resume the first meiotic division until a menstrual cycle. Therefore, remarkably, they all remain arrested for at least 12 years, and some as long as 50 years. In the first phase of the menstrual cycle, approximately 10 to 20 follicles begin to mature because of rising FSH levels. One of these follicles becomes dominant, allowing the primary oocyte to complete the first meiotic division. This division is asymmetric, leading to the formation of large secondary oocyte and a much smaller polar body. The secondary oocyte is released from the ovary and immediately enters the second meiotic division, but again arrests, this time in metaphase 2. The secondary oocyte remains arrested in metaphase 2 until fertilization takes place. Upon fertilization, the second meiotic division continues, producing an ootid and another polar body. The ootid quickly develops into a mature ovum or egg. Spermatogenesis is the formation of sperm. It occurs in the seminiferous tubules of the testes, beginning at puberty and continues throughout life. The process starts with spermatogonia, diploid stem cells that divide by mitosis to ensure a continuous population of precursor cells. These cells differentiate into diploid primary spermatocytes, which undergo the first meiotic division to produce 2 secondary spermatocytes, and the second meiotic division to produce 4 haploid spermatids joined by the cytoplasmic bridges. They subsequently separate from each other to become distinct haploid cells. Spermatogenesis takes about 2 to 3 months. Fertilization involves the fusion of gametes, restoring the diploid chromosome content. In humans, male and female gametes are usually brought together by sexual intercourse. Sperm travels through the cervix and into the uterine and fallopian tubes. Fertilization usually occurs in one of the fallopian tubes. Sperm are motile, but from studies of the time it takes sperm to reach the fallopian tubes, it appears that the female reproductive tract plays an active role in moving sperm to the oocyte, for example, through muscular contractions. In the female reproductive tract, sperm go through a key developmental step called capacitation. Capacitation is a series of physiological changes that allow the sperm to fertilize the egg. These changes include alterations in the fluidity of the plasma membrane, loss of some surface membrane proteins, and changes in the charge across the membrane, called the membrane potential. As a result, sperm show increased motility. Upon reaching the oocyte, the sperm passes through two layers before fusing with the plasma membrane of the oocyte. These two layers are an outer layer of follicle cells called the corona radiata and an inner matrix of glycoproteins called the zona pellucida. After passing through the layers surrounding the oocyte, the plasma membranes of the sperm and oocyte fuse. The oocyte then completes meiosis II, and changes occur that prevent polyspermy, or fertilization by more than one sperm. Although sexual intercourse is one way that gametes are brought together, a variety of other methods are also used to achieve fertilization and pregnancy in humans. Collectively called assisted reproductive technologies, these methods are typically used to treat infertility. For example, in vitro fertilization is a process in which eggs and sperm are brought together in a petri dish, where fertilization and cell division occurs. After fertilization, the single-celled zygote divides by mitosis, a process called cleavage. This process divides the single large egg into many smaller cells. In most organisms, these mitotic divisions are rapid, occurring at the same time, and are not accompanied by an overall increase in size of the embryo. Cleavage results in a solid ball of cells called a morula. Further cell division results in a fluid-filled ball of cells called a blastula. It is around this time, about 5 days after fertilization, that the developing embryo implants into the uterine lining, which is thickened and highly vascularized under the influence of progesterone from the corpus luteum. The pattern of cleavage is different for different organisms and is determined in part by the amount of yolk present in the egg. As shown to the left, animals with lots of yolk, such as insects, fish, and birds, produce embryos that have two poles, a vegetal pole where the yolk is concentrated and an animal pole where it is less concentrated. The presence of yolk inhibits cell division. Therefore, in these animals, cell division does not extend through the entire egg. In contrast, animals with very little yolk, such as mammals and sea urchins, cell division extends completely through the egg, as shown to the right. In the absence of yolk, the developing embryo needs another source of nutrition. In humans and other placental mammals, this nutrition is provided through the placenta. Implantation marks the beginning of pregnancy, the carrying of one or more embryos in the uterus. It lasts approximately 38 weeks from fertilization. Because the time of fertilization is often not known, human pregnancy is typically measured as 40 weeks from the last menstrual period. Pregnancy is divided into 3 periods called trimesters, each lasting about 3 months. The How Life Works text provides this visual synthesis that guides students through the 9 months of fetal development. I highly recommend that students review this resource. During the first trimester, the embryo develops all its major organs and begins to be recognizably human. Toward the end of the first trimester, a human embryo is known as a fetus. Early in the first trimester of mammals such as humans, the blastula has an inner cell mass and an outer layer of cells and is known as a blastocyst. The inner cell mass becomes the embryo itself, and the outer cell layer forms part of the placenta. The placenta is composed of both maternal and embryonic cells and is the site of gas exchange, nutrient uptake, and excretion of waste between maternal and fetal blood. The placenta also secretes the hormones estrogen and progesterone, which maintain the uterine lining during pregnancy. Meanwhile, as shown by the patterned arrow, the inner cell mass differentiates into a two-layered bilaminar structure consisting of the epiblast and hypoblast. Gastrulation is the second key developmental process in embryogenesis. It is a highly coordinated set of cell movements that leads to a fundamental reorganization of the embryo. In humans, cells of the epiblast divide and migrate inward. These migrating epiblast cells form three layers or sheets of cells called germ layers, which include the outer ectoderm, intermediate mesoderm, and inner endoderm. All of the adult tissues and organs are made from the three germ layers. At this stage, the embryo is known as a trilaminar, three-layered embryo to reflect the three germ layers. Organogenesis, the third key developmental process, is the transformation of the three germ layers into all the organ systems of the body. The ectoderm becomes the outer layer of the skin and nervous system. The mesoderm makes up the circulatory system, muscle, and bone, and the endoderm becomes the lining of the digestive tract and lungs. The second trimester, from around 13 to 27 weeks, is characterized by continued development and growth of the fetus. This is the time when fetal movements first can be felt by the mother. Because of improvements in medical care, babies born in the last few weeks of the second trimester often are able to survive. The third trimester, from 28 to 40 weeks, is characterized by rapid growth, with the fetus doubling in size during the last 2 months. Weight gain is critical, as birth weight is a major factor in infant mortality. Although 40 weeks from the last menstrual period is the average length of pregnancy, a baby born between 37 and 42 weeks is considered full term. Childbirth marks the end of pregnancy. It consists of 3 events: changes in the cervix to allow passage of the baby, descent and delivery of the baby, and delivery of the placenta. These 3 events result from strong muscular contractions of the uterus mediated by the hormone oxytocin from the posterior pituitary gland. During pregnancy, the cervix is a tube about 4 centimeters long, closed with a mucus plug. To allow passage of the infant, the cervix shortens and widens. These changes are not enough to accommodate passage of the baby during delivery. The head and shoulders of the fetus are wide relative to the size of the pelvis. The baby goes through a series of movements that facilitate its passage through the pelvis. Babies are usually born head first with the face pointing down toward the mother's back when passing through the birth canal. Other positions make vaginal birth more difficult and sometimes impossible. The third and final stage of childbirth is the delivery of the placenta, which usually occurs about 15 to 30 minutes after the baby is born. Although birth normally occurs vaginally, in some cases, such as when the baby is too large or positioned feet first, it is necessary to remove the baby by making an incision in the abdomen and uterus in a surgical procedure known as a cesarean section.